Hey gang, welcome back. This week we're going to be talking about the science of gains, right? Make sure that you have that Z on there because gains is life, bro. So we're going to be talking about resistance training, right? When we're talking about resistance training, and especially coming off of the endurance training section last week, we really need to highlight the two different types of training, right? Regardless of the style of training that you do, whether it's endurance training or it is resistance training, the muscle is going to respond and adapt to that stimulus. Our muscles are amazing at adapting. That's what they're there for. That's why the human body exists. We need to be able to adapt and respond. The major driver and the major difference between these two, while it's obvious, is going to be that the specificity of the training stimulus is going to actually be different. Go figure, right? Great example of this. 60 minutes of endurance activity. Think about how many skeletal muscles contractions you're going to actually have occurring, right? Think about how many times, let's say you're running for 60 minutes. Think about how many times your calves are going to contract, how many times your hamstrings are going to contract, your glutes, your arms, whatever, right? Like you can have 5,000 plus contractions in an hour for one muscle. That's wild. Let's flip the script and let's look at resistance training. Right? You're not going to have nearly as many contractions occurring when you're resistance training. You could have 20 to 30 in an hour. You could be doing a typical bro split, 3 sets of 10, hitting the buys and getting out of there. And you're going to do significantly less contractions. Even if you have a crazy hard workout, you're still going to have less contractions overall than you would if you were doing endurance activity. But the major difference is going to be that you have a high level of stress to that muscle. Like you're mechanically stressing that muscle in a different way. In order to really go into resistance training, we got to talk about three main terms. Well, really four. But the first one is going to be 1 rep max. And this is going to be our 1 repetition maximum. So that's the maximal load that you can lift through an entire range of motion, right? Like you got to actually squat to depth. I'm sorry, no quarter squats, they don't count. Muscular endurance is going to be the ability of a muscle to make repeated submaximal contractions against a submaximal load, or make repeated contractions against a submaximal load, right? How many times can you curl 20-pound dumbbells? Muscular endurance. The principles that we talked about in endurance training with overload and specificity, they're still going to apply here, right? They're actually going to apply a little bit more because we can have cross kind of training that occurs with running and cycling and stuff like that. Here, if we're only ever training arms, our legs aren't necessarily going to get stronger. So something that we need to talk about is how does the nervous system play a role in resistance training? We kind of touched on this earlier-ish when we were talking about the nervous system in general and really getting into excitation contraction coupling and how excitation contraction coupling in the movement across the neuromuscular junction is going to drive muscular contraction. Well, that doesn't just happen overnight, right? Like our ability to optimize our nervous system and optimize the way that our nervous system actually signals to contract during resistance training has to be trained. And in fact, the muscle strength increases that you see in really 2 weeks, but the first 2 to 8 weeks of taking on a resistance training program, that gain is actually going to be due to neuromuscular control. Like we're getting better at moving. It's not because we're getting bigger, right? We're getting better at that movement. It's completely different than the endurance training aspect of this, right? Remember we talked about endurance training, you don't have cross education. Here, we can actually get cross education. If you only train your right arm, your left arm will get stronger. You're improving the motor unit and like the neuromuscular continuum, if you will. You're essentially improving the coordination. That's the word I was looking for. You're improving the coordination. Jesus, Parker. So we can actually have cross education. So let's say that you get put into a cast. You break your elbow and you end up in a cast. Well, if you are able to actually train the opposite arm, the contralateral arm, you can still see increases in strength. Endurance training doesn't get that, right? So if there's such a high level of neural input coming in, then there's got to be a very clear pathway for how the neural signal is making it through. And essentially, anytime we're talking voluntary muscle contraction, right? Like I command the biceps to contract, right? Or if you're doing curls or you're doing leg extensions or squats or whatever, you're voluntarily contracting a muscle. And that initiation actually begins in higher brain centers. And it's going to be like that first perceived thought of, oh, I need to make this move. We're then going to send that signal to the motor cortex. Motor cortex is going to send that signal to the brainstem, which is going to run down the spinal cord out into the motor neurons, and those motor neurons are going to depolarize, hitting the muscle, spanning the neuromuscular junction, and we get contraction, right? So starting in the higher brain centers, moving in the motor cortex, sending that message down the spinal cord and out to the effector muscles, out to our target muscle contraction. That's how voluntary muscle contraction works. So this signal, this neuronal depolarization, or this neuronal signal, is going to be known as neural drive. And that's essentially a term that we can manipulate, and we can increase or decrease the magnitude of the neural drive. And one of the best ways that we can actually measure this is we put electrodes onto the muscle. So we actually do something called EMG, or electromyography, and we're actually able to read the electrical signals across the muscle. Just like we can do with EKGs, just like we can do with the heart, our skeletal muscles are muscles too. They function on electricity. We can actually read this. We can read the electrical activity. And then our body can actually adjust the magnitude of that signal based on how many motor neurons we're activating, the firing rate, how fast we're hitting those muscles with signal, the unit synchronization, the order in which we're recruiting muscle fibers, as well as the ability to get that signal across the neuromuscular junction. So we can tone up or tone down that neural drive. We can increase or decrease that neural drive in order to achieve the desired outcome. So if you go back a couple of weeks, we actually talked about this when we talked a little bit about skeletal muscle system, and we talked about fiber-specific tension. And essentially the principle that I want to bring forward a couple of weeks is that the amount of force that's able to be produced by the muscle is directly related to the number of actin-myosin cross bridges we can create. So if we have a smaller muscle, we're not going to be able to create as many actin-myosin cross bridges as if we had a big muscle. If we had a big muscle, we can create more actin-myosin cross bridges. So the whole reason that our muscle is actually going to adapt and grow is to increase actin and myosin interactions, because we need to be able to exert more force, and we need to be able to exert more force and more force and more force, because the more force we're able to produce, the less relative force that we have to use. Does that make sense? The more force that we're able to produce, the less relative force that we actually have to use. The stronger we get, the lighter relative a working weight is going to get. So what about type 1 fibers? So if we're going to be doing resistance training, does that just mean we're targeting type 2 fibers? Not necessarily, right? Like, we are going to target type 1 fibers as well. They're just not going to hypertrophy. They don't have the same level of satellite cells. They don't have this increase in mononuclear domain that we're going to talk about. But what we do, I said do, I can't say it now. What type 1 fibers do is they increase calcium sensitivity. So they're actually going to respond more favorably to released calcium at the troponin level. So if we bind troponin to calcium to troponin, conformational shift of tropomyosin, we allow for that strong binding site, actin-myosin, so we allow for the ratcheting to occur. We allow for the skeletal muscle contraction to actually occur. So how do we grow? How do we add actin and myosin units? How do we add or facilitate these interactions? Well, there's two kind of concepts here, right? We'll have hypertrophy or we'll have hyperplasia. And what hyperplasia means is it's the addition of muscle fibers within a specific muscle cell. So that is saying that, let's say we start with 100 muscle fibers, which is low, and we do a resistance training program and we end up with 120 muscle fibers. That would be hyperplasia. We've increased the number of muscle fibers there. By increasing the number of muscle fibers, we've increased the amount of actin and myosin cross bridges that can occur, right? Does this actually occur in humans? It's still up for debate, right? Like, is it logical to sit here and say hyperplasia occurs in humans? Yes, it is, right? The concept that we are born with a fixed number of muscle cells is a little bit hard to swallow. It's a hard pill to swallow. But does it occur in high levels? No, it does not, right? There's some human evidence to suggest that hyperplasia occurs in humans. There's some evidence to suggest that it doesn't really. We know in animals with extreme weights and extreme situations, we can get pretty significant levels of hyperplasia. So this one's open for debate, right? It doesn't mean that it's not going to happen, doesn't mean that it is going to happen. It's like saying that Hafthor and I have the same number of muscle fibers. Or when Hafthor looked like me, when he looked his best, he had more muscle fibers, or he had the same number of muscle fibers as he has now. I don't know, right? So if hyperplasia does occur, how do we actually increase the number of muscle fibers? Well, there's two general concepts. One, we actually kind of split those muscle fibers, right? Like you're tearing Velcro apart and we're splitting and kind of fraying it. And that allows for the increase in the number of muscle fibers. The second one is that we actually increase recruitment of satellite cells, and rather than just adding nuclei to the cell, we actually start forming new cells. Right? We don't know. End of story. We have no idea. But what we do know, What we do know is that hypertrophy does happen, right? And what hypertrophy is the increase in cell size, which is what we want when we want to do resistance training. Everyone wants to get big, but nobody wants to lift no weight. Ronnie Coleman. So essentially what's going on is that after several weeks of resistance training and several weeks of getting beat down, our muscle decides to respond and it starts to increase in size. And by increasing in size, we actually are going to increase the number of actin and myosin contractile units or contractile proteins that are able to be used. And what's really unique about hypertrophy is that essentially we're adding sarcomeres. So we're not adding cells, but we're adding sarcomeres, right? The Z disc to Z disc sarcomeres, just increasing contractile proteins. And what we see is that during hypertrophy, we don't add sarcomeres in series, right? So it's not like we have 1, 2, 3, 4. What we do is we actually add them in parallel. So we actually begin to stack sarcomeres vertically so that we actually get a growing or a widening of that muscle cell or of that muscle tissue, which is what essentially is hypertrophy, right? That's what we see when we see hypertrophy. We don't get really long muscles, we get bigger muscles, and that's adding them in parallel, not in series. Well, what's the time course really look like on hypertrophy and resistance training? You generally don't really see increases in hypertrophy until at least 3 weeks. Remember, the first 2 weeks is really going to be neural, 2 to 8 weeks really. The first 3 weeks, you might start seeing some increase in cell surface area, right? So in that CSA. So we might see increases in cross-sectional area, CSA cross-sectional area. It would make sense that because you have more contractile proteins, that you would be able to exert a greater level of force, right? Because we're getting bigger and we're gaining more contractile units, you would think that you'd necessarily get stronger. It doesn't always work out to be that way, right? Just because you get bigger, your muscles get bigger, it doesn't mean that your strength is automatically going to go up as well. There's a lot that goes into strength, right? Like there's neural signaling, so there's motor unit recruitment that's going to go on. There's also Golgi tendon organs, right? Like there's neural things that are going to actually adjust and serve as a governor based on the amount of force that you would allow your muscle to make. So there's more to force or more to strength than just size, unfortunately. That's why bodybuilders aren't necessarily the strongest individuals in the room. So we kind of alluded to this a little bit earlier, but how does resistance training affect muscle fiber type? Well, actually with resistance training, you would think that you would go from type 1 towards type 2 or type 2A towards type 2X, right? You would think that you'd get more fast twitch. And what we've seen is that that's really not the case. You're actually going to be moving closer to a type 1 fiber. So a training stimulus or training adaptation to any type of training is going to be the progressive shift away from a type 2X and towards this hybrid type 2A, type 1 type fiber. It's just what happens with training. We become less fatigable. We become more efficient, right? If you look over on the right, this is something that's called a Western blot. And what a Western blot is it's a biochemical technique that we can do to actually look at protein concentration. And essentially, what we're going to look at is the thickness of the band here. And the thicker the band, that means the more protein that is present. So if you look, there's actually a specific time slot or area in which you're going to have an occurrence of a type 1 protein or type 1 myosin ATPase. And that's going to occur in this zone right here. So as you look across, anywhere you see that banding, that means there's type 1 fiber present. There's type 1 myosin ATPase present. So that's why when we look across, we can see that there's a pure type 1, there's a hybrid type 1, type 2A, and then there's a hybrid that's got type 1, type 2A, and type 2X all in one muscle fiber. That same trend can be carried across for those type 2s, type 2As, type 2Xs, right? All we're doing is looking at the proteins. I'm not going to ask you a question about this on an exam. It's just a cool piece of technology that we can do to visualize changes in protein. So if we're improving our muscle, are we also going to be improving other things? Yes, like without a shadow of a doubt, by adding heavy stress to our body, by spinally loading or really loading our body in general, we're going to actually have to strengthen a lot more than just our muscle to respond to that stimulus. We're actually going to be strengthening our bone dramatically as well. We're going to be strengthening our muscles, tendons, but also our bone. We can see significant increases in bone mineral density as we increase resistance training. There's really nothing else out there like it. It is one of the best ways to increase bone density. So we're going to increase bone strength and decrease the risk of fractures by resistance training and improving bone mineral density. So let's kind of look at this a little bit more biochemically, and really it'll help us understand what's going on. So there is this concept that muscle protein synthesis and muscle protein breakdown are occurring. Essentially, we're anabolic or we are catabolic. We're building more muscle or we're destroying muscle. We're building more proteins or we're breaking down proteins. The more anabolic we are, the higher the level of muscle protein synthesis we have going on, the more protein that we're actually making. The more catabolic we are, the more muscle wasting we have, the more muscle protein breakdown we have going on. So essentially what we want to do when we're resistance training is we want to stimulate the maximal amount of protein synthesis that we possibly can. So what we know is that one single bout of resistance training can actually significantly improve and increase muscle protein synthesis, but it also significantly increases muscle protein breakdown, right? We're tearing the muscle. We are literally and physically tearing the muscle as we are doing lifting. As we are weight training, we are literally tearing muscle, right? So it makes sense that we'd have an increase in muscle protein synthesis and an increase in muscle protein breakdown. It just so happens that the recovery from the muscle damage, right, from the resistance training activity is heavily favored by muscle protein synthesis. So we actually significantly outweigh muscle protein breakdown with muscle protein synthesis following resistance training. Now, why do you not get big instantly? Essentially, this muscle protein synthesis time course, or the time with which our body has to be in a positive muscle protein synthesis state, is about 3 weeks before we'll start seeing significant increases in cross-sectional area. So it takes 3 weeks before you start seeing changes in muscle size. That's crazy because a single bout of exercise can increase muscle protein synthesis by as much as 150% for 2 days. The thing is, as we get more advanced in our training, that time course actually decreases. Our body gets more efficient and it gets used to the damage and gets used to the breakdown. So we're actually not in such a high level of muscle protein synthesis. So it could be one of the reasons for some of the noob gains, right? Like it could be that with training age, their training age being so low, their muscle protein synthesis rates get so high that they're able to actually put on muscle faster. As they get used to the training stimulus, it actually basically decreases the anabolic response to it in some way, shape, or form. It doesn't really, but it decreases the time duration. How is it that we actually gain muscle? How is it that we actually go through muscle protein synthesis? Well, that's what the next couple slides, I think it's the next 5 slides, are really going to be discussing. Essentially, there is a very specific set of steps or signaling events that occur within the muscle to actually signal to increase protein synthesis. So what's going to happen is we're going to get cell signals in the sarcoplasm saying, hey, I need to grow, I need more muscle, right? Cell signaling event occurs. we're then going to stimulate a muscle transcriptional activator, or something called a transcription factor. This transcription factor is a molecule that actually gets in and interacts with the DNA and actually turns on genes or writes genes. That transcription factor moves from the cytosol to the nucleus to do that, to write those genes. And how it's going to write those genes is it actually makes messenger RNA. Just like you learned about in high school biology, you're going to actually have a movement of this transcription factor into the nucleus. You're going to write DNA, and that messenger RNA that results is going to get sent out of the nucleus into the ribosomes. So it's going to send it back in the sarcoplasm to the ribosomes. That ribosome is going to basically take that messenger RNA, internalize it, think about it, and then say, oh, okay, I need to make a specific protein in response to this. I need to see what's going on, and I need to make this exact protein, right? It's that simple. A cell signal is received. It is that simple. A cell signal is received. Transcription factor writes the gene, right? So it's literally writing the gene. It's creating messenger RNA that's getting sent out to the ribosomes. Ribosomes are taking that, reading that message, and then creating a new protein. Could be actin, could be myosin, could be titin, could be any of these contractile proteins. Could be others, but it could be any of these contractile proteins that we actually need to increase hypertrophy. We kind of talked about all this already, but those ribosomes are actually going to be the individual things that are factory linking the amino acids together. So they're actually going to be compiling the proteins and linking the proteins together to make targeted protein. What's really cool about this process of hypertrophy and cell signaling and muscle growth is that resistance training by itself actually increases our ribosomes. So we actually, not only do we increase the contractile proteins that we need, we increase our ability to make those contractile proteins. It's crazy, right? Like it really, really, really makes sense when you start thinking about it. This is where it gets a little bit more complicated. What is the signaling event that is calling on the transcription factor? What is the preamble to this, right? Well, essentially, the very first signal that we're going to take and we're going to see is going to be a mechanical signal. It is going to be a stress that is placed on the sarcolemma. It is going to be a stress that is placed on the muscle cell membrane that actually stresses a mechanoreceptor to a level that which it says, oh, four-letter word, we need to adapt. This is going to get real fast. We need to come up with a way to adjust to this in the future. So we get mechanoreceptor activation, right? And what's really kind of cool is that it hasn't always been thought that it's this mechanoreceptor activation that's going to be driving most of these signaling events. What was originally thought is that it was actually hormone signaling, right? Like it was originally thought that it was IGF-1 or insulin-like growth factor 1 or growth hormone or some of these other anabolic hormones that are presented in response to heavy resistance training. But it's actually these mechanoreceptors. And it's going to be these mechanoreceptors' end goal to activate something called mTOR. And what mTOR stands for is mammalian target of rapamycin. And all that mTOR does is it essentially acts as a signal to grow. It is a cell growth signal, right? It is the master regulator of hypertrophy, actually. And there's two ways that mechanoreceptor is going to actually activate mTOR. It's going to use phosphatidic acid, which is PA, over here on the left-hand side of the screen, or it's going to use RHEB, R-H-E-B. Now, mTOR and phosphatidic acid, PA, and RHEB work in very funny ways, in really kind of, I think personally, neat ways, but really kind of funny ways. Essentially, when we get a mechanical stress, we activate levels of phosphatidic acid. We actually increase levels of phosphatidic acid. What is it about this mTOR that's so special? Well, it's going to increase transcription factors, and it's going to actually activate the transcription factors to increase the protein content. So mTOR is actually going to be located on a lysosome. You don't need to know what lysosomes do, but mTOR is actually going to be on the lysosome, and so are phosphatidic acid, and so is Reb. And at rest, there's low levels of phosphatidic acid on that lysosome, and Reb is going to be inhibited. So we're not going to get activation of mTOR at rest. In order to activate mTOR or get that mechanical stimulus to actually be signaling to mTOR, we have to do two things. We have to activate Reb, and we have to increase the level of phosphatidic acid. Literally, directly as a result of the mechanoreceptor, we're going to increase phosphatidic acid. It's going to be increased, right? Our muscle is going to send a signal saying, oh my gosh, we need phosphatidic acid because we got to tell mTOR to increase and we got to get more protein. The other one, right? So the Reb is a little bit different. So essentially how Reb gets activated is there is an activation of something called extracellular signal-related kinase, or ERK. The activation of the mechanoreceptors, the activation of these sarcolemmal signaling molecules is going to actually activate ERK. ERK is then going to inhibit TSC2, which is going to allow Reb to increase. And that increase of Reb is going to actually activate mTOR. So if we look at that here, we're going to actually have a change in TSC2. So we're going to essentially use ERK to reduce TSC2, which will increase Reb, which will increase mTOR. So just a little chance to geek out a little bit on sports nutrition. This target, this mTOR target, is something that is massively popular in sports nutrition. This is why we recommend taking protein after exercise a lot of times. This is why we recommend taking protein before exercise. This is why the protein guidelines are set the way they are for resistance training athletes. And this is why the amino acid leucine is such a hot topic and why people try to take branched chain amino acids, or BCAAs. There's research that shows that BCAAs or leucine or protein are able to actually maximally stimulate or significantly increase mTOR. What's interesting is, does that mean that you can just take protein, just take leucine, just take BCAAs and maximize muscle mass without exercising? And the answer is no. We have to have all of the amino acids present, and we have to have that mechanical signal in order to get the hypertrophy to actually occur. How long does this process take, right? Like, it seems like this would take forever. And realistically, within minutes of having our first muscle contraction, we're going to start to get mTOR activation, right? All we're doing is saying, hey, you go tell this person, hey, you go tell this person, hey, you go tell this person. It's not going to take long to get that signal from the sarcolemma to mTOR. It's just not. Within seconds, we're going to activate Reb and increase phosphatidic acid concentrations. And then within hours of the training stimulus, we're actually going to significantly increase muscle protein synthesis. So it just takes hours to get this muscle protein synthesis increase, but it takes a minimum of 3 weeks. It takes 3 weeks of being in a positive muscle protein synthesis state in order to actually get that hypertrophy. On the next video, we're going to talk about hormones, talk about their use, and then finish up the last couple of slides. See you guys in the next video.